Welcome everybody to our last session of the day, session 4, and remind everyone that afterwards we have a dinner starting later on this evening downstairs, and there will be ample time to continue networking amongst each other. And then after the dinner, the bar is open until late, so there's no limit to the bonds that could be formed at this meeting. Exactly. These are bonds that even force microscopy would have to confirm as being very solid. All righty. Well, our next speaker is Dr. Chris Brazel, to whom I once again extend my appreciation for shifting of your time. There was a very nice flow from Chris's presentation coming after Jim's presentation because Chris earned his PhD in chemical engineering from Purdue University. But Chris continued to move on and has now authored over 70 research publications, and his group regularly presents at national and international meetings in the field of nanotechnology. Dr. Brazel is currently a professor of chemical and biological engineering at the University of Alabama, which is the home university of our distinguished keynote speaker for tomorrow, Dr. Bob Borge. Chris Brazel's topic for this afternoon is nanomedicine for treatment and diagnosis of cancer, again, continuing an important theme that we've had throughout the day of medical reasons to develop advanced nanomedical technology. Dr. Brazel?
Thank you very much. Changes my introduction a little bit now that we've had the chance to have the break, but I certainly appreciate your call in letting us get refreshed a little bit, get the audience a little coffee, may make the presentation a little more interesting for everybody out there. I would like to thank Dr. Rothblatt for bringing together everyone to this meeting. It has been a very exciting, interesting time so far for me. I do need to make sure that I distinguish my university slightly from Dr. Borge. I thought you were going to say we were the home of the Crimson Tide football team. I'm at the main campus of the University of Alabama, which is separate from UAB, which has the medical school, although we're not that far apart, and we do find our ways to collaborate once in a while. Someone else mentioned that it can be fairly intimidating to come to a conference like this. A few years ago, I started in nanomedicine, so my body of research is much less than Dr. Leary's 30 years. You'll see when my title comes up, it's relatively long. I think it may be the longest title of any of the presentations today. But that just means my talk is a little bit more focused, maybe, and a little bit less of a body of work, so maybe it'll be a little bit shorter as well. So if you could start my talk. Oh, I've got this to do my talk, don't I? Thank you. Okay, I can start my talk myself. The area that I've been working in has been in drug delivery, and we're working on now some nanomedicine ways where we can try to do 4 different things to target, image, treat, and report back on whether cancer therapies can be effective. First of all, and I'm sure most everybody in the audience here will look at this chart and realize these are the major contributors to mortality in the U.S. and probably most developed countries in the world. I gave this slide a few weeks ago to somebody who's actually working in the cardiovascular area. They said, yeah, well, you're working on cancer, but number 1 is still heart disease. That's why I have this slide. Because if you look at heart disease, cerebrovascular disease, and some of the other ones, there have been significant improvements in those areas. This is 1950 versus 2004. Cancer has had very little change in the mortality rate in the U.S. over 5 decades, over 50 years. And there's still a large need then for cancer therapies that can be easily targeted and hopefully reduce some of the side effects that we're all familiar with for cancer as well. If you have anyone who's going to go to the hospital, of course you know the main routes of options would be surgery, chemotherapy, radiation therapy. Hyperthermia is much less common, although it has been around since the 1960s. And generally, if in most cases, as was mentioned earlier today, with the combination of radiation therapy and chemotherapy, combination therapy in nearly every case is much more effective. The patients recover much better, and we can treat and kill many more of the cancer cells. The goals of our work are to try to create a nanoplatform, something that can be easily addressed to certain cells within the body that can target, of course, be able to use for imaging, such as gadolinium, which is sort of, no pun intended, the gold standard for imaging, as well as treat, and then I'm going to add in a fourth component, which is also reporting back whether the cancer cells have actually been killed or not. We'd like to maximize the effectiveness of our treatment while trying to minimize the side effects. That's one of the main goals of drug delivery in any case. Try to get the drug exactly where we need it to go, reduce the total number of molecules we have to get into the body or the amount of therapy that has to be done. This is a slide from the National Cancer Institute that listed what their ideals were if they could have the best possible system that combines these four categories. They like to have targeting, imaging, therapy, and a reporter cell. And within this particular device that we're working on, it includes, of course, targeting, which can accumulate the drug or the therapy at a particular site, also avoiding the side effects to healthy tissue. We'd also like to include imaging, which can, of course, then allow perhaps for early detection, reduce the size that we might be able to see within individual tissues as far as to where these things localize, be able to do treatment. We'd like to, of course, stop tumor growth, perhaps even kill tumor cells, and use these combined mechanisms of action to get better therapeutic effects, and report back, was the cancer treatment effective or not? We're going to first of all talk about the targeting. In our case, we're looking at various vectors that can target specific cancer cells. I'm working with, of course, people at UAB. They're just down the road, and we've got some experts in targeting using an adenovirus. David Curiel and Micah Avarts are two of my colleagues that work in this area, and they have the ability to change what some of the amino acids are expressed in a region of the adenovirus capsid so that we can attach certain things to that. So they can express cysteine or histidine, molecules that then we could attach magnetic particles to, attach polymeric carriers, and use this as a way of targeting. We'll also look at other ways of targeting, perhaps using the traditional antibody antigen connection. We're working on nanoparticle design. I work with a team of chemists, and I've been learning a little more of this myself because my training is more in the polymers field. But we're working on nanoparticle design, and one of the things that we can do with magnetic particles, if you apply an alternating current field around them, the size of the particles and type of particles will tell us how much heat they can generate. So we can use a magnetic field to induce hyperthermia within the patient. This is an alternative photodynamic therapy that can perhaps pass through a patient much farther than the other kinds of infrared light that had been mentioned before. One of the issues with this nanoparticle design is something called the Curie temperature. The Curie temperature is the temperature at which the nanoparticle will stop heating and become the superparamagnetic particle will no longer have the behavior where it will heat. Most normal materials at the normal size, this Curie temperature is 800 to 1,000 degrees Celsius. Kind of cook maybe more than just the cancer tissues if we have to worry about temperatures that high. So one of the ideas that we've had is to try to work with new magnetic particles that we can reduce the Curie temperature that will cut off at a level that's safer for the patient. Keep it to where we get 40 to 45 or maybe 50 degrees Celsius at the local site, and the healthy tissues around it stay at normal body temperature. Hyperthermia, I should have mentioned that as well, is the idea that we heat tissues to about 42 to 45 degrees Celsius or so. Healthy cells can survive that temperature. They're not happy there. There's a lot of side effects. If the original work that was done with hyperthermia was whole body hyperthermia. You induce a fever about 108 degrees Fahrenheit, 42, 43 degrees Celsius. The cancer cells preferentially die off or stop growing, but the patient has so many side effects that therapy by itself is not used nearly as often. Or regional hyperthermia could work too, but still there's so many side effects involved. Now we're looking at magnetic fluid hyperthermia, and we're not the only ones. There's a number of groups looking at that, where we can try to treat cancer cells by localizing the cells first, the magnetic particles first, and creating the heat just where we need it. A number of magnetic particles also have the nice capability that can be used for imaging. So in addition to being able to locate and treat the cells through hyperthermia, we can image where they've gone. And because we're looking at surface characteristics of the cells, we can target them to even metastasized cancers instead of having to deal with whole tumors and treat the cancer very early on. The hyperthermia is done using an alternating current magnetic field, very similar to MRI, although we're trying to avoid using the same exact properties of an MRI machine, so a patient could still be imaged that way, but perhaps have a different device that ends up being developed, which can focus and maximize the energy we can get by placing these particles in the body. And then the other thing, and this is sort of where my expertise comes in, because I was trained in drug delivery when I was at Purdue, and we'd like to combine the therapy where we can do hyperthermia plus use a temperature, high temperature modulation to cause drug release, and we're using some phase-separating polymers that can do that. As far as doing the localization work, I mentioned earlier that we're looking at adenoviruses, although the NIH panels that review our proposals weren't too happy with adenoviruses, so we're switching, I think, more to antibodies now because of the 1 or 2 cases of adenoviral therapies that have gone awry. Either way, we can get to the cancer cells with different ways of targeting the cell surfaces. We don't really care in this case with the hyperthermia therapy whether the nanoparticles are pulled inside the material or not, because we can kill them effectively by basically thermally ablating the cell membrane. For the chemotherapy agent, we may be more concerned with actually getting the device across the cell membrane and to a particular area within the cell. But in either case, it's a huge improvement over traditional chemotherapy. I mentioned before that we're working with this adenovirus. This is just a portion of the adenovirus. This shows one of the knobs up at the top, and then the green section here represents what's called the hexon region of, in this case, the adenovirus we're working with. This is the part that we can easily genetically engineer to produce different peptides, which will allow us to attach other things. So we can put our magnetic nanoparticle there, we could put a temperature-sensitive polymer that encloses the drug as well, and have a linker molecule that will connect everything up together so we can make the whole thing eventually at the nanoscale. One thing different from earlier talks that I heard, it is certainly advantageous to work with materials that are already available. And some of the materials that are commonly used would be magnetite. Most of the folks that work in this field use that. I'm collaborating with a chemistry professor who's part of our Materials for Information Technology Center at UA. So we've got a number of experts in making magnetic nanoparticles, and he's very interested in trying out these different types of structures, which might give us some better properties. In some cases, we might be able to get the Curie temperature matched to 50 degrees Celsius or so we don't overheat the patient. In some other cases, for example, cobalt ferrite or manganese ferrite, these work very well as phase contrast agents, so we can use them for imaging on top of the hyperthermia therapy and get rid of the gadolinium. Not necessarily that bad, but we can do both of those things with the magnetic materials. And we've worked with a variety of different materials as far as which ones might give us the best amount of heating so that we can use the least amount of nanoparticles to still be effective. We had a custom-designed unit developed for our lab, which consists of a coil that we can surround our nanoparticles with. This is one that has the size that will fit in about a good-sized test tube. We also have one that's set up where we can test Petri dish type scale, and another one that's a paperclip coil, so we could actually use that one on animals through their skin. There's some advantages and disadvantages to each of those. But one of the things we're trying to investigate is how to optimize the amount of heating we can get into these materials. The hyperthermia chamber can go up to 5 kilowatts, and we can go through a range of radio frequencies from 50 to 485 kilohertz and try to optimize it for the different kinds of particles we're working with so we can maximize the heat. We're taking our images using an infrared camera. We found out very early on that a thermocouple, which is made of metal, tends to go haywire when you put it inside an AC magnetic field. This actually worked to our advantage because now we can actually see the profiles and see how uniform the magnetic field is. So this is a picture on the left here to begin with of where we start. You can obviously see where the coils are, and in between the coils is where our sample is. And when we turn on the magnetic field, we can heat very effectively. And this is showing a temperature of close to 60 degrees Celsius in our nanoparticles dispersed in water. And if we look at the graph over here, we can vary how much they heat depending on how many particles are there for one. This graph shows using different magnetic field strengths from about 100 to over 600 Gauss. So these are some parameters that we have that we can change around with and vary so we can optimize the amount of heat that might go into the device. I have a coworker in my lab, one of my postdoctoral researchers, who did some work at Yonsei University before joining my lab. And they'd done some work with mice, induced a skin cancer into these mice, and treated them straight with hyperthermia. They placed magnetic particles in each of these. You can see the one on the left has the tumor. That was the control in that case. The one on the right had the same tumor, but after treating with hyperthermia, returned back to fairly near normal. And we can see from the graph here, this also shows for the same experiment, the control and where we put magnetic particles but no magnetic field were the top two, and the volume of the tumor continued to expand. Whereas the third experiment, the one shown in purple at the bottom, the magnetic particles, when you apply a magnetic field, the hyperthermia was able to basically keep the tumor size the same during the process, so they didn't have any additional growth of the tumor. Along with doing any of this kind of work, I think the mathematical modeling and trying to understand what's happening is very important. One of the things that, of course, we want to look at is how much heat goes in and where does it go. If you think about tumor tissues or cells in the body and applying heat to any one area, how much heat do we actually need, and how fast does it leave that particular area? So one of the analysis we're doing is from the Penne's bioheat equation, and the heat terms that are involved in the equation include, of course, the change in temperature with time. How much does it heat up? And the heat that we place in the magnetic particles can conduct away through the tissue area, through the solid parts. We have a power generation term, which is our magnetic particles. And then blood perfusion is also important as well, because our entire bodies are perfused with blood. The blood moves through there fairly quickly, and it stays pretty well thermally regulated, so we're going to have a cooling term related to the blood perfusion. We'd like to, of course, be able to tune the Curie temperature so that we don't have this overheating where we might affect healthy cells or large areas of tissue outside of the region we're looking at. So the modeling we're looking at is either with regular nanoparticles or perhaps with the low Curie particles. We could see if that's even important for the analysis we're doing. One of the early pieces of work that my master's student had worked on was assuming that we had a tumor region that had nanoparticles dispersed throughout it, and then a healthy tissue region right next to it, of course. And we could apply a magnetic field and hopefully optimize the temperature into a therapeutic region where the tumor is, but on the right side, we could reduce the effects to the healthy tissue and keep it closer to 37 degrees. And this showed that there would be nice ways where we might be able to heat just the tumor region and avoid some of the side effects. Oh, this didn't show up with everything. Well, the one that's important is there. The first one's a flat graph because we started at time 0, but this shows kind of the generation of heat during the hyperthermia process, and this is all from the mathematical modeling. And we could see that we can raise the temperatures quite rapidly at the center or the core of where the tumor was placed, but it does cool down fairly rapidly, and this is from taking analysis of fairly common convective heat transfer, so that we can be fairly certain, even if we can't control the Curie temperature fairly well, that only the region where the nanoparticles are should be heating to a very high degree. Along with doing this, one of the things that we, of course, can optimize is the nanoparticle concentration. We could also look at optimal particle size. The particle size, and this is really where nanotechnology comes in, will be optimal for different types of materials. We've done some calculations for manganese ferrite and cobalt ferrite and found for cobalt ferrite the optimal size is right around 10 nanometers. That's where the resonant frequency can heat most effectively. For manganese ferrite, it's a little bit lower. I believe it's around 7 nanometers. So for each of these different particles we're looking at, we're hoping that the system that we have, we can figure out which particle size we're going to need to get effective hyperthermia treatment. Another thing we're very interested in is understanding where the nanoparticles go, both for toxicology as well as if we just place this against cells, where are they going to behave? So this is actually work from an undergraduate student that's in my lab, but he was able to attach a fluorescent dye into our particles, and we're getting ready to do some confocal microscopy work as well to figure out where perhaps the nude or the magnetic nanoparticles would go on their own versus once we get the attachment to the different targeting vectors. But this is all things that are still in the future in our lab. If we switch to looking at drug delivery, the idea of putting a drug in the body and having it have a long-term release has, of course, been around for some time. It's much more challenging to put the drug in the body and tell it now, release. Find that exact time and find some triggering event where we can get the drug to come out exactly when we want to. If we're trying to do cancer therapy, we don't want to inject and have the drug to come out until it's localized. So if we look at triggering events, pH is used quite commonly. Most of the oral medications we have, of course, a pH dissolvable gel on the outside, so the drug releases in the intestines and it's most effective. We could also, of course, use temperature and some other things that we could use to play around with these neat polymers that can phase separate and release a drug when these things change. Some of the options mentioned earlier, we could use infrared or light energy. That can certainly pass through the body. It's a nice, non-invasive way where we can try to trigger the release or some event that is going to cause drug release. This was some of the early work in this area was done back in the early 1990s with photoinitiated reactions using angioplasty. And last fall, I actually taught heat transfer, and I was very excited to see that there was an example in my book, which is a junior level class, of photodynamic therapy for treating cancers. So my undergraduate students are even starting to see some of the nanotechnology come out. The option we're looking at is magnetic field, but most of the early work with magnetic field and drug release was done for localization. And there's even some of that that's being done now, using a stationary magnetic field to try to localize particles in a certain area, such as the blood-brain barrier. But there have been a lot of problems with getting very effective drug delivery in that way. And if we go back again to the mid-1980s or so, Bob Langer's group at MIT was doing a lot of interesting work in drug delivery. And they had some initial ideas of using a magnetic particle, and if you apply a field, it'll move around a little bit and squeeze out some drug. It did, but it was a millimeter-sized magnetic bead, and it didn't squeeze out a whole lot more drug than if it wasn't there. So we think that perhaps now with the nanotechnology, we can get much smaller particles. And instead of using the magnetic field directly to do the drug release, what we're proposing is to do magnetic field combined with thermal delivery. So we use the magnetic field to create the temperature to cause a change in the polymer, and then release the drug that way. The last way up here on my slide that I have for doing the drug release is electronic. Many patients have glucose or insulin delivery pumps that are implanted. There you have something that has to have an interface outside the body, and it's fairly invasive. The concept of doing what we call magnetothermal delivery, of course, is going to be some sort of injection and localization to the tumor. I've asked my students to replace this fairly sophomoric picture of the human. And we're not necessarily targeting to the appendix. Anyway, it does get the point across. We're going to use traditional methods of antibodies or adenovirus, perhaps, to do the targeting, and this, of course, through the cardiovascular system. Once it gets to the site, we can apply an external magnetic field. We could apply it to the whole body in this case. The AC magnetic field will not heat up healthy tissues where there's no magnetic particles, and we can treat anywhere the particles are. We do have some concerns if the particles all end up in the liver and we thermally ablate the liver, even though it's not cancerous, and we'll be working on these issues, certainly. After we apply the magnetic field, of course, the particles heat up, and as they dissipate, we're working on designing a system that can phase separate and release the drug. One design for the system includes a grafted polymer system where the grafts work kind of like molecular gates. At a low temperature, they close off the drug release. When they heat up, they open up, and the drug, which is the little red sphere, starts to come out. And then over time, once the magnetic field is turned off, the gates reclose. Any drug remaining stays inside. So we could use this perhaps for multiple pulses of releasing a drug. Although, for the most part, at least our initial work has been on larger scale polymers that have magnetic particles in them. We are looking at, and I think my next, slide after this. We are looking at some nanoscale whole platforms now, which is going to be kind of the next generation of this. As we do drug delivery, one of the things we're really interested in is trying to get a phase-separating material that's going to release the drug rapidly when it's heated. And if we look at the systems we've worked with, PHEMA, which is the first system, is basically the same material that's used in soft contact lenses. It's biocompatible, it's a good material, but it's not thermally sensitive at all. And the higher the bar is, the faster the release. So we'd like to have the red bar, which is the highest temperature, release fast, whereas the blue and green bars release very slow. So we've had to switch the system around from a thermosensitive gel to a grafted gel. And this was a proof of concept that we could at least get a change in the amount of release so that it releases faster when it's heated. We've gotten many more results since then. I just didn't have the latest results from my graduate student for this slide, where we've changed the structure of the material so that going from 37 to 45 initiates the drug release. Here it was between 25 and 37, just because that was an easy proof of concept. But now we've gotten up to where we can trigger it from body temperature to slightly higher, which is our hyperthermia temperatures. And with the grafted gel, it will work to some extent at least. It's not as good as the opposite. If we could chill the body down and use hypothermia, that would work great, but I haven't found a way to do that yet. We're working on a way where we can now move into doing hyperthermia initiated, or not hyperthermia, the drug release initiated by the magnetic field. So we have a flow-through system that we've set up, fairly rudimentary, but we can actually place our magnetic materials inside these coils and connect everything up through a flow system and look at the drug release initiated by the magnetic field itself. The other system we're working on now is micelle-based. And that's where a lot of folks are working also on nanomaterials, because micelles are one of the natural nanoscale materials. In our case, we're working with a meltable micelle. And we're working with polyethylene glycol, which is the corona area, the blue parts, and then polyecaprolactone will actually form a crystal that melts at about 40 degrees Celsius, which is ideal for us. So the challenge for us is going to be able to get magnetic particles inside this micelle along with the drug. And when we heat it, the crystals melt away, and the micelle hopefully will also be able to deliver the drug. A lot of this is still in the future. We're still getting all of our ideas together with where we can go. Of course, imaging has already been brought up. These are not pictures from my lab. I do collaborate with Tian Ng at UAB, who does experimental MRI, the very high frequency, high magnetic field MRI, where we can get the ability to get very fine resolution. But perhaps the next slide is more interesting. This is some work that Tian Ng's group did at UAB, looking at some tumor tissue. And the nice thing now is we can combine MRI with traditional NMR. And you'll have to trust me that there are actually lines on there that look like NMR, but they're labeled. And the important thing from the graph here is that we can detect an NTP, which is nucleotide triphosphate, or ATP. So we could figure out, as we move across the image, whether the cells are living or not. If there's no ATP, we've killed the cells. This is the reporting mechanism that we thought when we first saw that slide from NCI. How in the world? We can go in and we can make sure we release the drug, we can get the particles attached, we can image where they are, but how in the world do we know that it really worked or not? And that's, I think, is still a big challenge for a lot of the kinds of devices we're trying to use in medicine. But we have high hopes that the experimental MRI might be a way now where we can figure out if the cells we're trying to treat actually do have either a decrease or perhaps no ATP being produced, which would indicate that our therapy has been effective. And that's it for my talk. I have a number of collaborators. Of course, my collaborators at UAB may expand now that I'm meeting Dr. Borge. And this is my research team that involves a number of graduate students and undergraduate students and postdocs.